if you take a look at this has been an exercise that went on in several countries, and basically estimates are not incredibly different. And the second thing which is striking is that if you sum up all these numbers, there is a 40%, okay? And 40% is basically a kind of every cancer epidemiology that works in cancer prevention. We basically are confronted to these estimates. So the proportion of preventable cancer cases is 40%. It is incredibly difficult to go beyond that. Nutritional epidemiology, the context is incredibly complicated. We work with diet, of course. This is what we eat, but then foods, they come with nutritional content that is incredibly important because they give us information about the biology of food and how they interact with their host factor. There are factors related to what we drink, of course, alcohol, but also to our lifestyle. Tobacco, obesity is a new epidemic in several parts of the world, and of course, physical activity has been well. Some people they really think that physical activity is the one. I mean, apart from smoking, of course, but it is the number one suggestion that for cancer prevention that one could give to the general population. The environment, of course, is incredibly important. And many different facets of that, and there is growing and if not established evidence that metabolic health plays a role in this context. So I probably don't need to spend a lot of time to persuade you that nutrition is incredibly complex. It's difficult to isolate components because we do not tend to do one single thing or eat one single thing. There is a lot of issues related to exposure classification. When it's down to behavioral sciences, confounding and selection bias are there, and then we need to deal with that. We have identified associations between dietary components and cancer risk, but very often we do not understand the mechanisms, basically why we observe these associations and why certain dietary components or lifestyle components, they are carcinogenic. There is, of course, a lot of endpoint heterogeneity in cancer, unlike other non-communicable diseases, and very often we deal with small effect. So what are the perspectives? Shall we give up investigating the link between diet and cancer risk? I ask myself this question probably every day, and I still found good reasons to do that, but I think the answer is not necessarily obvious. We know that diets contribute to cancer risk. A lot of it is probably through obesity and metabolic dysfunction, but there are very many aspects of diet that we can think they influence cancer risk, but it's so incredibly complicated to understand them and investigate them in observational studies. So what can we do here? Well, there have been many significant recent technical developments that made it possible to integrate several types of exposure assessments into observational epidemiology. And I'm talking primarily about the availability of molecular measurements. So GWAS, this goes back 20 years, but then there are new acquisition techniques to generate molecular data. I'm thinking about proteomics, metabolomics, epigenetics data. And there have been also progresses in the analytical frameworks to assess causality. So it's very important that we use any weapons, is a bad word, apologies, we have to investigate these relationships in a very challenging domain. Triangulate evidence is very, very important. So IAC is the International Agency for Research on Cancer, is a WHO cancer agency. We have a mission, and it's basically the first one is to describe the occurrence of cancer. The map I show in my second slide is coming from one of the branches here. We understand the causes of cancer. We try to understand the causes of cancer, and this is where my branch is pretty busy with. And then we also implement preventive strategies. And in general, what we do is we promote global collaborations among researchers, hopefully for reasons that will become clear in the course of my presentation. So I want to spend probably 1 minute describing the nutrition and metabolism branch that I'm leading. So this is a multidisciplinary environment. We have expertise in cancer epidemiology, in biostatistics, and also in laboratory facilities. And why is that? Well, basically because one of the domain, or one of the studies that we coordinate is the European Prospective Investigation into Cancer and Nutrition. Probably I don't need to spend a lot of time describing this, but this was a multicentric European study that was initiated back in the 90s that involved 10 countries and 23 administrative centers, as you can see in the map. And one excellent idea that researchers had back then was to collect biological samples that is currently still stored in liquid nitrogen at IARC. So this is a little bit the design of the study. You could see it started in the 90s. Dietary and lifestyle questionnaires were collected, blood samples was collected, and then there were follow-up questionnaires that made available some longitudinal data to be used in etiological analysis. Cancer ascertainment is happening through follow-up. I don't know about Mexico, but Europe, we are experiencing very huge problem about data protection. GDPR regulation by the European Commission has made our life incredibly complicated, particularly for scientists that would like to work in international context. So the large amount of biological data, of course, justify the investment into laboratory sciences. So my colleagues Sabina Rinaldi, Pekka Keskirakkonen, and Chrysovalantu Katsoyannis, they are leading laboratories within the branch, and you could see that there are various techniques going from the quantification of targeted assays for hormones and inflammatory markers and fatty acids to the left-hand side of the slide, and moving to the right, we go more into untargeted applications, and so for measuring polyphenols together with targeted proteomics through the Olink platform, and going down to the liquid chromatography mass spectrometry to measure untargeted metabolomics. Of course, this is a variety of techniques that render possible the generation of an increasing amount of molecular data that is used in our study. So this is a little bit our context. So we have a set of exposures in the black, in the yellow box that we can relate to the disease outcomes in the gray box. And then we could also appreciate or estimate or evaluate the role of, well, you call them either modifiers or mediators, very often molecular data. And then these analytical frameworks has been used to investigate kind of mechanisms. And so we have a lot of dietary factors, of course, as well as lifestyle factors and biomarkers measurements by means of metabolomics, proteomics, epigenetics. We also try to have research on microbiome. It's not obvious because we don't have fecal samples, at least in EPIC. And of course, our interest focuses on several cancer sites, but also cardiovascular disease, type 2 diabetes, and other non-communicable disease. All-cause mortality is also in our agenda. So I want to give you two or three examples of studies that we conduct. So using the same analytical framework, this is a study on healthy plant-based diet. So last year, Reynalda Cordova with Heinz Freisling here in the picture, they have found an inverse association between a healthy plant-based diet and cancer, type 2 diabetes, cardiovascular disease, and multimorbidity. This was a very fine study, and we're quite happy. But then we thought, okay, can we understand a little bit what is the biological pathway linking healthy plant-based diet with diseases? And so we used proteomics, and Peggy, Lair, and Heinz are working on a fairly complex analysis that I'll be happy to guide you through. So the healthy plant-based diet is a diet characterized by a large amount of vegetables, fruits, and legumes, of course. And then we had proteomics data that could inform or potential links on these relationships, because they could inform about a number of biological processes within our body. So this is going to be probably a little bit technical, but I try to guide you through that. So we have within EPIC, in a case cohort sample, about 12,000 studies participants with the 7K SomaScan proteomics data. Basically, there are 7,000 aptamers that inform about proteins available in 12,000 participants. And then we run relatively complex analysis. So in the first step, we related the proteomics data to the healthy plant-based diet. And then we identified signatures of plant-based diet. And the signature was made of 1,200 proteins. And of course, this was a big number. So we thought, okay, how can we screen them? So we used the CISPQTL, Decode Genetics, which is basically an online catalog. Well, basically what they do is we use genetic information that was strongly associated to proteins, and just what we want is to screen out those proteins that had strong instrumental variables, so that they showed a strong link with the proteins. And of course, we lost about 2/3 of the proteins, because for the majority of them, we didn't have a strong enough instrumental variable, because our final objective was to run a Mendelian randomization analysis, whereby we could use genetic data that are available to relate proteins to specific disease outcomes. So we were left with 67 proteins. The next step was to run colocalization, and then eventually we could identify 9 proteins. Colocalization is a type of analysis whereby we have a look at the genetic region that is related both to the disease of interest and to the protein of interest. And if the two are the same, we can say that the specific SNP, that the protein and disease, they colocalize if they have a common genetic origin. And so this is a strong evidence for causality. And so among the different and the 9 proteins, I like to focus on 3 of them. The CCM2 protein was associated to colorectal cancer risk. And then, of course, this makes sense because this protein is responsible for angiogenesis, is oxidative stress, and also is involved in inflammation processes. The NCAN type 2 diabetes link also made sense because the NCAN protein is a marker of metabolic dysfunction and is associated to steatotic liver disease. And this last one is a PCSK9 that was associated to cardiovascular disease. Well, the PCSK9 is a protein involved in LDL metabolism, and this is even a target for potential pharmacological studies. So this is a ongoing studies, but that I think what I'm trying to show here that we are using some of the newly generated molecular data to inform on diet-disease link and see whether we could identify candidate and sensible molecular pathways that could further be investigated. I move now on to the second example is ultra-processed food and cancers. This is a study that was led by my colleague Inge Übrichts, and basically relating the diets following the NOVA classification of food processing was linked to the risk of cancer. So basically, participants were ranked into minimally processed food, NOVA 1, some processing NOVA 2, processed NOVA 3, and ultra-processed food NOVA 4. And there were substitution analysis that were implemented. So what we found, apologies if this was a little bit busy, but on the left you can see different cancer sites and then associations between the 4 NOVA classification and cancer risk. And you could see that there were several inverse associations for NOVA 1, suggesting that a diet that doesn't make use of food processing was inversely associated to risk of several cancers, whereby the NOVA 3, we observe several associations for NOVA 3 diets and several cancer sites. And then the NOVA 4 was associated to head and neck cancer. Of course, we were quite surprised to observe these associations, particularly NOVA 4 and head and neck cancer. And what we did next is Inge and Heinz, they worked on a paper that was published last year. No, actually, this was back in 2023. But we were relating the ultra-processed food to cancer, to risk of cardiovascular disease and type 2 diabetes. So what I want to suggest here, show you here, is that positive associations were observed for the 3 disease outcomes. And this is the black square. But then when they adjusted for BMI, therefore checking how much of this association was going through the obesity pathway, you could see that there was very minimal change in the risk estimate for cancer, some change for cardiovascular disease, and of course larger change for type 2 diabetes, because obesity is such an important risk factor for obesity. These studies, at least for cancer, suggest that the adipositive hypothesis on why ultra-processed food might be associated to some specific cancer is possibly quite weak and could at best explain 20 or 25% of the whole relationship. That's why we have a study, and we develop an application that was submitted to the World Cancer Research Fund. And this is a study where we are hoping to investigate the candidate mechanism of carcinogenesis in the associations between ultra-processed food and risk of breast and colorectal cancer. And this is something that we developed together with Martin back in February, and now we are awaiting a response from WCRF. Alcohol, this is again another map. This is the proportion of cancer attributable to alcohol in different parts of the world. Of course, this reflects largely different prevalence of alcohol in different parts of the world. There is another heterogeneity element, which is alcohol doesn't necessarily influence all cancer sites the same way. Alcohol was associated to cancer of the upper aerodigestive tract, female breast, colorectal cancer, and liver cancer. But then there were questions whether there were associations with less established cancer sites. So 10 years ago now, I started a large collaboration with Dr. Stephanie Smith-Water at the Harvard T-Chan School of Public Health. And we were using resources of Diet and Cancer Pooling Project that she coordinates at Harvard. And so we focus on cancer sites that have an inconsistent prior evidence with alcohol intake. And at AYA, we focus on upper aerodigestive tract cancer in never smokers, pancreatic cancer, and thyroid cancer. So you could see here, these are all studies that we just published. We were able to confirm an associations between alcohol and UADT cancer in never smokers. Actually, the associations were very similar in never smokers and smokers. We found a positive association in pancreatic cancer, and also an inverse associations for thyroid cancer. I'm showing women here, but it was very similar in men. We have not necessarily great ideas on why there is an inverse association. And the inverse association is also confirmed for kidney cancer and non-Hodgkin lymphoma. We try to expand a little bit this work and see whether we could identify a biomarker of alcohol. And this was a collaboration that we established with colleagues at NCI. So what we did was we used untargeted metabolomics with a fairly complicated multi-tier design in EPIC and the ATBC cohort. And we could identify actually two features that were associated to alcohol intake. You could see the dose response curve of one of them. So one feature, we couldn't annotate that back then. This was 3 years ago. But the first one was the 2-hydroxy-3-methylbutyric acid. And then the good thing about this, well, I don't know whether it was a good thing, but the interesting feature of interesting element of this study was that, unlike alcohol intake, this feature was strongly associated with hepatocellular carcinoma in EPIC, pancreatic cancer, as well as liver cancer in the ATBC study. And this was quite surprising. So this feature has been observed as associated to alcohol in previous cohorts before. And I need to spend 30 seconds to say this. So this study started as a biomarker discovery study, but then we realized that probably what we have identified was not necessarily a biomarker of alcohol intake. It was more something that was informing us about the metabolism of alcohol intake and possibly potential mechanism, because the 2-hydroxy-3-methylbutyric acids is linked to rate of hepatic glutathione synthesis that it can increase in response to oxidative stress. And oxidative stress, in fact, is one of the candidate mechanism for alcohol carcinogenesis. So what we did that, and this is my last slide before the concluding remarks, is that we try to validate these in intervention studies. The women alcohol study that is led by colleagues at NCI. And so they enrolled 51 women. There was an 8-week treatment period with a crossover design. and that women were given different doses of ethanol, well, never exceeding 30 grams per day. So we confirmed the results of the 2-hydroxytrimethylbutyric acids, and also 45 other metabolites were identified. Ethyl glucuronide is quite well-known biomarker, but also the ethyl alpha glucopyranoside, they showed very strong association with alcohol intake. So now the ball is back here, because with the data we have and the nested case control study, we could check the associations between these features and cancer risk. Yes, I would go to the concluding remarks here. Is nutritional epidemiology generated quite a lot of evidence on the role of diet and nutrition in cancer development, but this evidence is not necessarily always replicated. And if we take a look at the table from the WCRF of the different dietary exposures and different cancer sites, in fact, the table has many different gaps, and I think it would be good to know more about all these gaps. That are incredibly new opportunity provided by omics data, both for biomarker discovery, but particularly for identification of biological pathways. Of course, the need of replicate findings, triangulate findings, never trust your results, always try to replicate, if you can, also with intervention studies. I don't think what we did was correct. Usually what you want to do is do an intervention study first and then replicate the findings in an observational setting. We did the other way around, but it is what it is. I didn't talk about that, but there is a great need to translate what we find. But there is also a lot of input. I loved Martin asking Abraham about, hey, you are the new generation. We want to generate leaders. I think leaders need to have rigor and be creative. This is the secret, I think. And of course, collaborating with smart people is essential. You can find the references of some of the things I told you today in a paper that was published in April in JNCI monograph. I need to thank a few people, Elmira, Diana, Peggy. They were busy with some of the analysis here. The colleagues in the nutrition and metabolism branch, of course. I don't know whether Isabella Ormier is there, but anyway, she was heading this branch 10 years ago, and we are still thankful to her contribution. A lot of good people in EPI, colleagues at NCI, and of course in the Talent Cancer Pooling Project. And with that, I thank you for your attention.
Muchas gracias al doctor Pietro por su presentación. Fue sin duda muy interesante. Y con esto nos acercamos ya al cierre de nuestro evento conmemorativo. Para ello le pedimos al doctor Layún nos pueda acompañar para dar las palabras de cierre. Antes de eso, solo me va a tomar un momento para dar 3 mensajes. El primero es que terminando el evento, tenemos una cita para tomarnos una fotografía en la escalera cósmica. Entonces, si nos ubican a los que hemos estado en algún momento aquí, pueden seguirnos para que sepan exactamente dónde va a ser y podamos tener un registro de esta celebración. El segundo mensaje es, maestras, desafortunadamente no tuvimos mucho tiempo para preguntas, pero sin duda queremos escuchar de ustedes, ¿no? Yo he visto sus caras y sus reacciones a lo largo de la presentación y seguro tienen muchas ideas y sugerencias para nosotros y queremos nutrir lo que hacemos a partir de sus observaciones. Así que en la revista que tienen en su bolsita de regalo vienen nuestros datos de contacto. En estos datos de contacto también viene el correo se revisa diariamente, entonces si tienen sugerencias, dudas, comentarios, nos los pueden mandar por esa vía y se los vamos a agradecer muchísimo. Y el tercer mensaje es que además de la labor agradecerles la labor que han hecho como participantes del estudio, quisiéramos también agradecerles su labor docente. Esta ha sido muy importante. Yo soy hija de una maestra normalista, entonces he sido testigo de primera mano de la entrega y el compromiso que tienen las maestras con sus estudiantes, ¿no? Entonces es una labor muy, muy hermosa la que ustedes realizan Y también siendo nosotros personas que hemos tenido oportunidad de estudiar una maestría, un doctorado, seguro podemos platicar con algunas de ustedes y decirles aquellos maestros que nos marcaron, ¿no? Y que nos incentivaron a seguir estudiando. Así que me gustaría agradecerles por esa labor tan importante que realizan. Y con esto le paso la palabra al doctor Layo. Adelante, doctor.
Realmente muchísimas gracias al doctor Pietro Ferrari. Nada más quiero resaltar que es muy importante escuchar lo que se está haciendo en otros países, lo que se está haciendo en la Agencia Internacional de Investigación en Cáncer, porque nos ayuda también a pensar dónde está la contribución de ESMaestras en el avance científico global, ¿no? Y entonces, era muy importante que estuviera aquí el doctor Ferrari. Nosotros hemos entablado desde hace muchos años una larga relación con la Agencia Internacional de Investigación en Cáncer de la Organización Mundial de la Salud y nos ha dado también la pauta a pensar, bueno, este estudio tan ambicioso que se hizo en Europa, nosotros tenemos un estudio así de ambicioso en México. Y entonces realmente estamos muy agradecidos, doctor Ferrari, muchas gracias. Nosotros recibimos también una felicitación de la directora de la agencia, la doctora Elizabeth Weiderpass, y esto nada más es una manifestación de cómo realmente ESMaestras está ya aportando a algunas de las iniciativas de la Organización Mundial de la Salud y que seguiremos aportando hacia adelante. Entonces, queríamos darles una probada de hacia dónde vamos nosotros en ESMaestras, al menos en el caso de cáncer. Muchísimas gracias, doctor Ferrari. Eh, y no quiero terminar sin decir que, pues, compartirles un sentimiento que tengo. Es un sentimiento de frustración. Es un sentimiento de frustración porque no tenemos suficiente tiempo para hablar con ustedes ni hablar con todas ustedes. Ustedes son las que nos acompañan en línea y nos acompañan aquí, pues son realmente una muestra pequeña de las 115,000 maestras. Queremos tener más tiempo para escucharlas. Esta frustración de la que hablo no es solamente mía, es de todo el equipo de SMAES, de investigadores del Instituto Nacional de Salud Pública, porque nos damos cuenta que hay mucho que aprender de su vivencia, mucho que aprender, digamos, de los datos que nos dan, pero también de la parte más humana de lo que es ser docente, de la parte más humana de lo que es ser mujer en este país. Entonces, pues tengo esa frustración. Ahora, también creo, afortunadamente, hay caminos para canalizar esa frustración y realmente este evento, estos 2 días, especialmente el día de hoy, nos motiva a nosotros para realmente encontrar la forma de acercarnos a ustedes. Yo creo que esta experiencia, como bien lo planteaba la doctora Carolina Pérez, nosotros que nos dedicamos a la actividad científica tenemos una responsabilidad muy importante de acercarnos a las comunidades que se van a beneficiar de la investigación que estamos haciendo y tenemos que ya hacerlo parte y hacerlo centro de nuestra labor. Tenemos que empezar a hacer investigación centrada en las personas, centrada en las comunidades. El labor que ha hecho con este evento Laura Quesada, todo el equipo de organización de este evento, creo que nos ha reforzado y subrayado la importancia de tener una coparticipación y hacer esto un trabajo de grupo con ustedes en el centro y con sus comunidades y con sus familias en el centro. Entonces, pues es un compromiso que nos llevamos. No sé si hoy, como el doctor López Ridaura que hizo promesas importantes el día de ayer, yo puedo prometer que vamos a hacer un esfuerzo más concentrado en estar más cercanos a ustedes. Creo que el evento de celebración de 20 años, al menos este componente del segundo día con ustedes, creo que lo tenemos que hacer todos los años. Y tenemos que ver la forma de llevar este evento a los 12 estados. Entonces, yo me llevo de tarea en encontrar el mecanismo para hacerlo. Pero bueno, y pues sí quiero agradecerles a ustedes, y también quiero, pues les pido a todo el mundo un aplauso para todo el equipo que hizo posible estos 2 días. este, Teresa Telles, que debe estar por ahí en algún lado, Laura Quesada, todos nuestros estudiantes que participaron en esto, realmente les agradecemos muchísimo. Esto ha sido realmente una experiencia muy, muy grata para todos. Y bueno, pues ahora sí los invito a la última parte del día, que es la foto para el recuerdo. ¿Ok? Muchas gracias. Entonces, ¿quién nos...